Okay, so our talk today is going to be split into several parts. So I'm going to talk to you about the more experimental side, and I'm going to talk to you about understanding synaptic mechanisms, first of all, in a basic biology setting, and then how we can apply this understanding to predict or understand some disease state. In particular, I'm going to be referring to dementia. And then my colleague, Mathieu, is going to take over and talk about how we modeled some of this experimental work and the way forward next. So the interest of my lab now, not jumping to the next slide. Oh, here we go. So the interest of my lab is looking at synaptic mechanisms using various techniques. And as I said, the first part is going to be about looking at the local circuit connectivity, in particular, neurons that are of inhibitory nature. So inhibitory neurons are very special. They are a small population of cortical regions, but they are very diverse. And I'm going to be referring to some of those, and I'll take it slowly. And then we'll talk about what happens to them in some disease settings. So the first part, as I said, is going to be on the basic science, and then we see how we can apply this further. So these are some of the reconstructions of the inhibitory cells in our lab, and yes, of course, they look beautiful. What do they actually do? So it's really important to have a multidisciplinary approach to studying cellular circuitry or any other aspects of neuroscience. So in our lab, what we try to do is collaborate with various types of people. Now, we incorporate behavioral studies and to looking at cellular circuits and then at a synaptic level. And we also collaborate with molecular biology. And of course, not to mention the computational aspect. So this is really important to us to have various different techniques in order to answer a question. So the main question I'm going to be focusing today is first of all, the mechanisms of synaptic communication between the excitatory and inhibitory microcircuitry in cortical regions, in particular the hippocampus. And as you know, the hippocampus is very important for memory and learning. And it's one of the first regions to be affected in neurodegeneration and such as Alzheimer's disease. So we will then jump to the second aspect, which is investigating the alteration of the synaptic mechanisms that I will discuss in Alzheimer's disease. Now, the techniques that we use, as I mentioned, involve behavioral studies, so looking at for example, cognitive deficits or anxiety. We're very much interested in mental health and Alzheimer's disease. And we also follow this up with in vitro electrophysiology combined with neuroanatomy and molecular biology, not only at a single cell level, but we're also now developing cell cultures from human stem cells from dementia patients and also control patients. So to go into the electrophysiology aspects a bit further. Most of you know, if you're interested in looking at functional aspects, that we do need electrophysiology, in particular, also patch clamp techniques to look at the functions of neurons, to look at the function of networks if we use more than one electrode. So this techniques allows us to have a reader, a biophysical reader of the neurons, and it simply involves, I say simply, it involves patching onto a cell with a glass micropipette that has the same intracellular solution or composition of the neuron, and it becomes one with the cell, and so we can have a readout. So we can look at the way it behaves when we pass current through it or voltage, depending on what question you have. We then combine this with neuroanatomy. So we use double immunofluorescence and we use confocal microscopy. We can also do that on prior to recording or after recording, depends what the question is. Now, say if we record the cell, we want to obviously identify the cell. So we must recover the cell using a horseradish peroxidase reaction, or HRP, and reconstruct the cell anatomically, and that's what they look like sometimes. So those are the primary focus. As I said, we do use other techniques. So using these techniques, we have found very interesting findings in terms of short-term synaptic plasticity. Now I'm going to focus first of all on short-term synaptic plasticity, and then see how then the modeling aspect will come later. So I just want to give you a snippet of an example situation or a problem rather than give you a seminar on a particular, a broad problem. So for example, we used mathematical modeling to look at short-term synaptic dynamics. So from the experimental side, we know that pyramidal cells, and this is an actual pyramidal cell in CA1, can exhibit several different types of short-term synaptic plasticity. Now, this is what I've done a long time ago during my PhD, and it was the first lab to actually report before some very famous people can also show aspects that a single axon from the pyramidal cell can exhibit different types of patterns of glutamate release. So if this green is the schematic of the axon of the pyramidal cell, and it targets one particular type of inhibitory cell, let's call this the OLM cell. In fact, it's a very specialized cell, the CA1. It shows this type of synaptic release pattern. So these are short-term synaptic dynamics. So it releases glutamate, and glutamate gets more and more bigger as the stimulus increases. So this is referred to as synaptic facilitation. Very important in timing these different dynamics. Now, the same axon from this neuron targets other different types of inhibitory cells. Here we have a basket and bistratified cell. Just as an example, and the difference between these cells is how they look, where they target. So these are schematic of the axon. So these target different regions of the pyramidal cells. So that's how they're classified according to the way they look, the way they fire. Now we have a separate talk on these. So just to say that if this axon targets this group of cells, the dynamics will be completely different. So here we have short-term synaptic depression, that the glutamate each time after the presynaptic decreases. So Henri and various other people have shown that these dynamics are very important in regulating spatial temporal coding. So lots of modeling has been done since the 1980s rather, and this has opened up a quite big field. So looking at these dynamics, this is from an excitatory cell. It was of interest to me to look at whether similar patterns exist at inhibitory cells. And we looked at connections amongst inhibitory cells. So this is again schematic between two inhibitory cells. These are all inhibitory cells. They're very specialized because they can either contact each other or other excitatory cells. There are some inhibitory cells that only contact their own kind, the inhibitory cells. And indeed, we find that they display specific inhibitory short-term synaptic dynamics. Now, just to remind you, these are inhibitory postsynaptic potential. So this is on the current cloud, that's why they're going downwards. And if you look here compared to here, it might look similar, but actually the timing is totally different. So in short, we see a very distinct spatial and temporal difference according to cell types. So these are inhibitory synapses, so unitary inhibitory synapses that are highly specialized. So this is all very interesting because it tells us something about the code. Now, this is again some examples, real examples of neurons that were connected with each other. So the red one is inhibitory cell, and this dark blue and the light blue is also dendrites and axon. They are connected with each other, and the immunofluorescence show that these were the recorded cells with ANCA, and they're positive for cholecystokinin or CCK, negative for another protein, always with double immunofluorescence. And so here we have the presynaptic cell-firing action potential, and this is the postsynaptic voltage response. And we see that it shows very nice synaptic facilitation. So this is just to show you an example of what we do and the difference in the dynamics. We see that the timing of when the presynaptic cell comes in is very important and very relevant because it would determine the amount of depression of facilitation or even augmentation. So one synapse, especially if it's inhibitory, can show different types of dynamics. So when people just show two pulses and call it synaptic facilitation or depression, this is a little bit inaccurate in my opinion, because you really need to see with train of response. This is a dynamic system. So in short, we have specific cell type short-term dynamics. We then wanted to look at how this is different for a tonic and basic inhibition. Now I'm going to show you an example of CCK cells and how we explored this further and how we investigated some particular autotype receptor or CB1 receptor that regulate inhibition. So again, this is just an example of a published work. If you're interested, please look at it further. So we find that inhibitory cells, which are very specialized and short-term dynamics, have a very specialized way of controlling when neurotransmitter is released temporally and spatially. So if we look at just in CA1, we see that there's a lot of chaos in the way it releases the neurotransmitter. But before that, if we focus on the black trace, this is control, long, slow response. This is all GABA A, not GABA B. And this slow inhibition is referred to as tonic or background inhibition. Now, this inhibition was actually suppressed just as a background, because when we apply AM251 to the bath, after 20 to 30 minutes, we see the inhibition or the tonic inhibition revealed and it gets bigger. So that means that these receptors were constitutively active. Now, what is AM251? This acts on cannabinoid type 1 receptors. As I said, cannabinoid type 1 receptors are very important in regulating synaptic or the level of neurotransmitter at the presynaptic terminal. So this is why we chose to look at this as an illustration. And we see that there's a huge asynchronicity of release. Now, we want our synapse, we used to think that synapses are very precise in the way they release neurotransmitter. And this is true if it's a albumin positive neuron. And Hefton Honast actually showed this, and we're the first to show the difference in synchronous and asynchronous release in the dentate gyrus. So we see similar findings in the CA1, and this is actually governed by CB1 receptors, which is a very interesting finding, and it's demonstrated here. So we find that the synchronicity of release, that's how the latency, which is the time when the stimulus is elicited from when the neurotransmitter acts on the postsynaptic receptor. That's what I'm referring to when I say latency. So latency is different in control conditions, but when we use AM25, which is an inverse agonist of CB1 receptors, we see more order. And if we use an agonist of CB1 receptor, we see again disorder or flexibility. And again, if we block the intracellular calcium or chelate the calcium with a fast-acting EGTA or BAPTA, it also brings some order, because that prevents the CB1 receptors from working because they need calcium. So these results highlight the importance of how systems like the CB1 receptor can regulate inhibition or the release of neurotransmitter. This is just a summary of what I've talked about so far. So at the center of the schematic of a synapse, we know that we have different types of short-term synaptic activity. And if we focus on inhibitory cells, we see that CB1 receptors really do regulate the way the neurotransmitter is released in their flexibility. Now, why do they do this? There must be some sort of reasoning behind this. And we think that when there is increase in postsynaptic activity, there's a change in the CB1 receptor involvement. And this has been published, and many others have shown this. Now, this allows the network to have flexibility, to allow some signals to go through and others to be retained, if you like. And it's very difficult to show this at an experimental setting. And this is why we need mathematical modeling. So, and how do these occur? So there's so many different proteins. And it's different to pinpoint pre and postsynaptic proteins simultaneously in experimental settings. So Mathieu will talk about how this has been modeled further. So knowing this knowledge, knowing the short-term, the complexity of synapses, we wanted to take this further and understand the process of neurodegeneration, for example. So we have a very nice AD model. So we started to look at Alzheimer's disease. The first question, we know that Alzheimer's disease is correlated with accumulation of amyloid beta and tau proteins, and this is correlated with cognitive deficits and neurodegeneration. So the first point is to investigate pre- and postsynaptic mechanisms that cause neurodegeneration. It's a big question. So we started to investigate this, and of course, what's the ultimate goal here? We know that there's no targeted therapy to alleviate cognitive deficits or even mental health problems associated with AD. So our ultimate goal or motivation is to design better targeted therapies to alleviate cognitive deficits and also psychiatric conditions. And that's the focus of my laboratory at the moment. And again, just want to emphasize why we chose to study Alzheimer's disease in particular. Well, it's the most common form of dementia, and 1 in 3 people will die of dementia. And AD being Alzheimer's is the most common form. And you can see from some of these numbers, I'm not going to go through all this slide, but you can see from some of these numbers that dementia is a global problem. It affects, in the UK alone, 24 billion people, which is more than cancer. However, there's such a disproportion of people who work in the dementia field compared to the cancer field. So this is one reason why we chose to study this. And another reason was that there are some really fantastic preclinical mouse models of Alzheimer's disease around the time I switched to the field, which was around about 6 years ago. So Japanese collaborators, they developed various lines of preclinical mouse models that accurately recapitulate the disease conditions in human beings. So one model that we use which is the APP knock-in mouse model that has 2 mutations or human mutations of the APP gene. And the second model that we use harbors the amyloid beta or APP gene as well as the tau protein. So the take-home message really is, without giving you more detail of the mouse models, if you're interested, please do look at these work or our previous papers. The nice thing is that it shows a really nice time-dependent pattern of the pathology shown here. And it's not an overexpression system. So it's an accurate or a faithful reproduction of what happens in the human disease. So we age our mice up to 24 months, just like here in these illustrations. So some of the data I've been going to show you do come from a very aged mouse, and I'll talk to you about some of the technical problems. So one other thing we did, as I mentioned, some molecular biology. So we looked at Wnt signaling proteins, which are Wnt signaling proteins, and these are presynaptic or postsynaptic. Now, presynaptically, these proteins are important in synaptic maintenance. Postsynaptically, Wnt signaling is important for postsynaptic receptor clustering and, for example, glutamate receptor trafficking. And we find that the Wnt signaling is different in Alzheimer's disease mouse compared to the kind of wild type control. And this has been published in Petraki et al. 2019 cerebral cortex. We find that from an early age, it's a 2 months mouse, which is equivalent to, let's say, a 20-year-old human being, that the Wnt signaling is already at a low level. And this is correlated with what we see from our recordings. So we recorded wild-type mice in AD model at different time windows. We have 1 to 2 months, 4 to 6 months, and 10 to 18 months as an example here. And we looked at the lateral cortex as well as the CA1 and the neocortex. And we see here from the black and red traces, if we can focus on this first, here these are synaptic responses, these are spontaneous responses. We see that there's more excitability in the AD model. So system is almost it almost looks like an epileptic system. And there are a lot of similarities there with epilepsy. So this epileptic seizure type activity or hyperexcitability starts from an early age, which is correlated with the dysfunctional Wnt signaling, if you like. And this then spreads to different cortical regions at different times. So it starts off in the entorhinal cortex first, and then it spreads to CA1 and then the neural cortex. And again, this is published work. Now, this pattern follows the human studies done from fMRI scans where we see high metabolic activity initiating the entorhinal cortex, which then spreads to different brain regions similar to our findings here. And this is from real patients who have cognitive deficits. So this is a really nice illustration of our model. And so the question is what to do further with these. And why this is the question here, the next was, how does the hyperexcitation occur? So coming from the interneuron background, inhibitory background, we looked at markers such as GAD67 and GAD3. So I'll go through these slowly. So GAD67 is a marker for GABA because it's an enzyme that produces GABA. And GFAP is a marker for astrocytes. And astrocytes are very important for maintaining our extracellular environment or neuronal health, if you like. So they're like the janitors of the system. GAD34 is a GABA receptor transporter, and this is specific to astrocytes. So here, these illustrations were done in images were taken in CA1 and also dentate gyrus of the wild type and the APP NLF mouse models of the AD model. Also, we've done similar studies in human confirmed cases of dementia patients, and we get similar finding. And the take-home message here is that there's an increase in GAD67. So there's more GABA transporters, and this is all correlated with more astrocyte activity. And when astrocytes increase, or reactive astrocytes and microglia increase the activity, is also referred to as neuroinflammation. So increase in neuroinflammation is correlated with more GABA production and transporter for GABA. So this is true for CA1 and dentate gyrus. And we also see that there is a selective destruction of inhibitory cells. Now, you may think there's increase in GABA. Yeah, there's selective destruction of inhibitory cells. This seems to be really cell type specific. So this illustration here shows you just the markers for AD, that it increases with age, as I showed, suggested with the mouse model, and Saito showed that as well. And we see that amyloid beta penetrates some cell types and not others. So it seems to be not affecting interneurons that are carotene positive. You can see that from the graph here. But other cell types like cholecystokinin and somatostatin, there seems to be more A-beta, intracellular A-beta inside these neurons. And there's a decline of these cells. There's more cell death in somatostatin and CTK cells. Why these carotene cells? Because they survive the system. So we see cell type specific inhibitory neurons that survive. Now, going back to our short-term synaptic plasticity, because that's where I started, we haven't quite got to that stage where we're looking at the unitary connections, because again, these are at 12 months. So there's that technical challenge of recording cells from double recordings, doing double recordings. So it takes more time, but that's something we're actually doing now. And so just to recap what I've said, we have neuroinflammation in our AD model, as well as accumulation of A-beta. And this is associated with selective destruction of specific types of inhibitory cells. And ultimately, this disrupts the excitation inhibitory balance. I've shown that there is hyperexcitation in the AD model. So the next question really is, what happens at that synapse, at the inhibitory level? This is something that we're currently working on. We're looking at different GABA receptor subunits as well, and how they are associated with this. So there's actually a paper that just came out today looking at background tonic inhibition. This is from our lab, and explaining some of these scenarios in Frontiers in Pharmacology if you're interested. So my colleague now is going to talk to you about some of the modeling that we really do need, because as you can see, the system is complex. There's so many complex things happening at a presynaptic and postsynaptic level. It's difficult to fully incorporate this in one experiment. But the beauty of mathematical modeling is that we can simulate these. And I am going to now pass you on to Mathieu. But before I do, I want to just take the opportunity to thank my lab and also my funders. So for this project was funded especially by Alzheimer's Research UK and Alzheimer's Research UK, UCL Drug Discovery Institution, especially Paul Whiting, who was a big supporter. And these are some of my students who did the work, Anna Islam, Wikong Sang, Richard, and then Alexander Petraki. So Mathieu, I'll pass it on to you. I hope I haven't overstepped my timing.
No. Okay, so let me share my screen. And I can get going. Thanks, Afia. And before I start, I would also like to thank Patricia for the kind invitation and the other organizers, you for listening, and apologies for not being there. I'm talking from Sophia-Antipolis, where the weather is rather bad today. Anyway, so this part is about the modeling, and I'm not going to talk about Alzheimer's disease. There's very interesting findings and data that Afia is obtaining and gathering, and we're starting to work on this. But what I will present now is step 0 that we have done some time ago, related to this differential release of neurotransmitter and the control of timing between pre and postsynaptic activity, and that was the startup of our collaboration. So the starting point of this modeling study is a very famous model of vesicle release and synaptic activity, synaptic transmission by Sodix and Markram. This model has been mentioned a few times already during this conference, and it's been very popular model for many reasons. First, it's in simplicity. It's a rather simple phenomenological macroscopic model, and also it can reproduce a lot of data. It can be fitted to a lot of data. This model is initially two-dimensional. There's two variables, D and F, that represent roughly for one, the number of vesicle or the occupancy of the release pool, and the other related to the release probability. So in absence of any perturbation or signal, incoming signal, I of t, this model is at rest. However, when an input spike arrives at the presynaptic neuron, then the interaction between D and F occurs, and the output, the interesting output of the model, which corresponds to the, let's say, the amount of neurotransmitter being released, is related to the product D by F. So like I said, this model has been used quite a lot. It's been derived in the late 90s, and it could reproduce a lot of data, showing in particular facilitation and depression that Afia has explained way better than I would do. But one thing that is not incorporated in this model due to its simplicity is the possibility to control the timing of release. So in this model, the transmission of the signal from pre to postsynaptic is instantaneous. However, it's not instantaneous in real life. There is some, at least minimal time for the neurotransmitter to travel from one side to the other. And it can also vary from simplicity configuration to configuration. Yes. Someone talked? Okay, maybe I'm hearing voices, sorry. So let me continue. The timing may vary from cell to cell and configuration to configuration. It's not captured by this model. Besides, due to the simplicity, again, there's no account in this model for the very complex biomolecular machinery at subcellular level on the presynaptic side, and that's the part that we were interested in following discussions with Pierre on this asynchronous neurotransmitter release that she had observed and recorded, in particular in inhibitory synapses. So talking about that biomolecular machinery that corresponds to what happens on the presynaptic side, there's a lot of work by many people for a long time, but In particular, we focused on the work by Thomas Südhoff and collaborators, who's done really enormous amounts of discoveries in this system, and in particular, he was granted the Nobel Prize for some of these results in 2013. So I will certainly not attempt to describe precisely this very, very complex cycle for the neurotransmitter release. I mean, it goes far beyond my poor knowledge in biology. But one thing that at least I can explain is that these different ways that this cycle can go and different timing of the release of neurotransmitter depending on the type of neurons has been analyzed by Südhoff and collaborators, and they could answer, at least in parts, some important questions that was animating this community about the possibility for shared mechanism of different mechanisms accounting for different modes of release, and they identified a shared mechanism that could be responsible for different types of neurotransmitter release, in particular identifying the key role of two families of protein complexes, SNARE complex and the so-called SM proteins, which really play a pivotal role in organizing these different types of neurotransmitter release. So that's our starting point, and we wanted to come up with a simple, parsimonious mathematical model that would reproduce at least part of the biological model of Südhoff, where SNARE and SM, these two protein complexes, would be our main dynamic variables, state variables, and we took into account other groups of proteins, which there are many of them, that have a sort of regulatory role in these complex cycles, and consider them as parameters that could allow us to go from one scenario to another. Okay, so of course we can come up with mathematical equations that do something and produce a nice output that qualitatively would resemble some experimental data, but maybe more interesting, also more complicated, is to try to also provide, at least in part, some justification for the forms of the equations, the number of variables, and the nonlinearities that we use in this minimal model. So we've tried to do that, and we managed, again, at least partially, to justify the model, which was inspired by some classical models of protein-protein interaction in the context of a cell cycle. In particular, we inspired by work from Tyson and collaborators, and came up with these rather simplified but still complex schematics that take into account a number of these biomolecular reactions, organizing the binding and unbinding between SNARE and SM proteins. So that's a little bit about the derivation, starting from this type of mass action laws that are legions in cell cycle models and protein-protein interaction. We use some consideration inspiring from the work by Südhoff and collaborators to reduce a little bit this model and identify two main variables which will play the role of the SNARE and SM protein complex respectively. And very importantly, as I will explain a bit later, the key aspect of the model is that it has multiple time scale, because what turns out to be, at least loosely speaking, an important point of this SNARE-SM interaction is this possibility for delayed interaction. So there is some binding and unbinding between these two, let's say, protein complexes, but somehow we can say that there is some sort of inertia, and this inertia will translate into a delay in the way this incoming signal is transmitted to the rest of the system. So our approach to model this delayed transition was to think of multiple time scale and the interaction between slow and fast variables. So with a little bit of work, we came up with a minimal two-dimensional model that would have multiple time scale due to the presence of a small parameter here, and that would react to an incoming signal V of t that will correspond to input spikes. We added to it an additional structure that I will explain a little more in a moment, related to some excitability of the model, and which will be related to the role of calcium. So it's important to say that calcium is very important in this cycle. We chose not to model it precisely because we wanted to focus on the obtaining a simple mechanism for the delay. However, we have a sort of mechanism that is related to the action of calcium, and I'll say more in a moment. Okay, so that's the complete model. It has 3 blocks, so to speak. The middle part is the classical Sodix-Markram model with these D and F macroscopic variables for the occupancy of the release pool and the release probability. And the big add-on, the novelty of our work, is this first part of the model, P1 and P2, that describes the interaction between SNARE and SM proteins with a different time scale that will kind of account for the inertia between them in the way they will bind and unbind. And finally, we can couple to electrical activity to obtain some postsynaptic response, and possibly, depending on what we put in there, some even spiking activity on the postsynaptic side. Okay, so that's the object of study, but for the rest of the talk, I will focus mostly on this P1, P2 part of the model, because this is what we brought in this study. Okay, so here again, this is the form of the system that we obtained from a little bit of work and inspiration from this cell cycle model. And on this cartoon, what I'm trying to do is, and that also summarizes the thought process that we've been through to try to organize the model and the different parameters that we needed to match with the biological model by Züdov. I superimpose the sort of space of configuration or phase plane P1, P2 of this part of the model to a cartoon showing the different stages of the vesicle cycle. And by really trying to decipher the different stages, we came up with the necessity of having a number of states in this model. In particular, we have a stable equilibrium S, which is the starting state that will receive the perturbation, so to speak, the input signal that corresponds to the incoming spikes. And there's also another state U that is an unstable stationary state of the model, which plays the role of a sort of energy barrier, saying that upon the arrival of an input, if the input is sufficiently strong, then we will cross this barrier and then enter into the next phase with this point, special point TC that I will describe next, and that will be the machinery organizing the delay. We have also a number of parameters in the model, and that we have tried to relate these parameters to families of additional proteins, these regulatory proteins that I told before, in such a way that we could identify different regimes, in particular 3 main regimes, 3 main modes of neurotransmitter release that can be encapsulated in this minimal framework. The so-called synchronous release, where the timing is roughly within normal bounds, a few milliseconds. The asynchronous release that Afia has mentioned in our part of the talk, where the release takes longer to occur, and also the possibility for a spontaneous milli-release that are also sometimes observed in absence of incoming spikes. Okay. So before I move on to further describing this P1, P2 model for the SNARE-SM protein interaction, I would like to take a few minutes for a small interlude on an important part of mathematical theory that is used a lot in modeling, also of course in theoretical studies, but that has proved over the years very useful in modeling, and that is related to bifurcations. So I know that in the room and online, there's maybe a majority of modelers, mathematicians, physicists, computer scientists that probably know about this very well. But I think the point of this meeting is also to discuss interaction with biologists. We have one next to me, well, virtually next to me for this talk, and I hope a few more online or in the room. So I would like to say just a few words about what this bifurcation theory is and how we can exploit it when we do modeling. So effectively, if we have a model with at least one of them, but at least one relevant parameter, we can, of course, analyze this model, sometimes with pen and paper, and very often with the aid of computer simulations. When simulating a model for certain values of parameter, we obtain a trajectory. We can visualize this trajectory in time, which is probably the most natural way to connect to an experimental time series. But we can also see this trajectory in the so-called phase plane or phase space projection, where we plot one variable against another, which gives additional information about how the trajectory evolves from an initial condition and possibly, in this case, converge to a stable equilibrium, a stationary state. So there are important long-term dynamical states like stable equilibria, like on this cartoon, but there are also in models unstable states. They might not be observable in real experiments, but nevertheless, they are important because they mark boundaries between different regimes. In particular, in the context of neuronal excitability, some type of excitability rely on the existence of a stable state and also a nearby unstable state in such a way that if a perturbation is applied to the stable state, then if the perturbation is strong enough, then that can elicit a spike before the trajectory converges back to the stable state. And the idea is to escape the basin of attraction of that stable state that is given by precisely the unstable state. Okay. Another way that is interesting when we have a model is to look at what happens when varying parameters. And in particular, if there are changes in the type of solution that we obtain. So here in this figure, you see that for different values of the parameter alpha, we may obtain a convergence to a stationary state, or for higher values of alpha, a convergence to a periodic state, so a rest state or a spiking state, say, in the neuronal context. And it's interesting to identify the transition between these two regimes, which in the mathematical terminology would be corresponding to what we call a bifurcation point. This diagram here is a sort of synthetic way to summarize this information. There's a regime here, which is a stationary regime. There's a periodic regime, and at the boundary between the two, there is a bifurcation point. This particular example is called a Hopf bifurcation, but sometimes it's also called a Hopf-Andronov Poincaré bifurcation. So a little wink to Henri in the middle of my talk. But importantly, these elements give information about the long-term dynamics of the system. So we take an initial condition and we see what happens after sufficient time has elapsed. Here we will have a solution converging to a stationary state, and here to a periodic state. But when we are trying to relate to experiments, it's also very important to understand transients, and long-term dynamics may not be so relevant depending on the context. And this is where there is an interesting add-on to bifurcation theory that is named dynamic bifurcation theory. And it gives rise to these delay effects that are really at the center of the modeling approach that we have proposed for these differential neurotransmitter release. So let me explain a bit more. So you take this diagram that we had before, where I suppose it's an applied current was a parameter. And now, instead of a constant or step current, we consider a slowly varying current, let's say a slow ramp of current. Well, what would happen in the model is what you see here. There is no stationary point or purely periodic solution anymore, but more dynamic transitions. So in this region, you have a quasi-stationary behavior. In this region, you have a quasi-periodic behavior. However, interestingly, there is a transition between the two, as you would maybe guess from what happened in the scenario where I was a parameter, but there is a delay. That transition occurs with a certain delay. This is maybe at first quite surprising. It's a well-known phenomenon in mathematics, and it's been explained and used in a number of models. In particular, there are big parts of computational and mathematical neuroscience where this type of dynamics is exploited. If you model bursting oscillations, then effectively in a lot of these models, what you have is a number of these delayed transition or dynamic bifurcation. One that would explain the transition from quiescence to burst, and another one that would explain the reverse transition. Okay, so there is this phenomenon, and this is due, loosely speaking, to the fact that now I is not a parameter anymore, it's a slowly varying quantity. So we have an extended model with multiple time scale, slow and fast processes. And that induces delayed transition between different regimes. If you have another bifurcation, so this is another example of bifurcation called transcritical bifurcation. That's a very simple model that would display this bifurcation. So no oscillations here. You have two stationary states, one stable and one unstable. And if you move a parameter statically, you see that these two stationary states, they get closer and closer to each other. They merge at this particular value of lambda, the parameter. Then they exchange the relative position and stability. So the one that was stable becomes unstable and vice versa. Now, if you play the same game as before, instead of having a static parameter, you consider a slowly varying quantity, then what you will observe is you don't have stationary points per se anymore, but you have a dynamic transition through this bifurcation. And there is, again, a delay. And one can understand intuitively this delay in the sense that the fact that the parameter is now a slowly varying quantity introduces a contraction of the system in this region, and in opposite there is an expansion here. And one needs to understand that the total contraction accumulated here, like a spring with some memory, would have to be balanced by the total expansion that you have on the right hand side in order for the trajectory to react and go away. So this is again a change of stability with a delay due to the fact that we have a slowly varying quantity. And in this case, we have a way to control somehow the timing of this event. And the underlying mathematical machinery that does that is the so-called Wayne-Weyl function. So that's what we have in this model. That's the scenario that we chose because we found that it's interesting and possibly to relate to the underlying biological phenomenon. So we have two variables representing this interaction between these two protein complexes, and they have different time scales. So there's one slow and one fast, and we have a delayed transcritical bifurcation. Okay, this is a rather dry slide, but in a couple of slides, I will give you a better account of what this model does and how to relate to the interesting phenomenon of in mind biological phenomenon. Regarding this Wayne-Weyl function, there is a way to evaluate the timing at which the instability is felt by the system. This is related to the evaluation of a certain integral, which one can do analytically or numerically, depending on the system. And we can use that to control precisely the timing and moment where that input signal will be transmitted to the rest of the system with a certain delay, and pair that with, for instance, some parameter fitting in the rest of the model. In this way, for every synaptic event, we can control the delay according to the experimental time series that we want to fit, as well as the other parameters of the model. Okay, so a little more about this P1, P2 some part of the model before I show you how it performs with data. So this is again the phase plane with these different states that we need, the stable state, the unstable state, and then this dynamic or delayed transcritical bifurcation. And this is what happens now. If there is an input signal that perturbs an initial condition at the stable state, mimicking an input spike that arrives to the presynaptic terminal, then sufficiently strong input spike will cause the perturbation to cross this sort of energy barrier and land on the other side of the unstable equilibrium, where it can enter into the regime of dynamic transcritical bifurcation. So in this first phase, which relies a lot on the fact that this one of the two variables is so, then somehow the input spike has no effect on the interaction between the two main variables, this SNRS and proteins. However, after some time has elapsed, which we again can control, then the effect of the input spike is felt and the fast dynamics kicks in, which effectively gives us a way to have a delayed response to this input spike through the interaction of between P1 and P2. And that delayed response will then be passed on to the vesicle release part of the model, the Sodix-Markram model. So that's the main machinery that organizes to this delayed release. Okay, So I'm still doing good on time. Of course, we have focused a lot on trying to match as much as possible the mathematical equations and the machinery that we used to the biological model, with the aim, of course, to test this model against data. We've tried data from inhibitory synapses recorded by Afia in our lab. So this is an example where we can see that calibrating the model for each release event allows to capture both the behavior of the postsynaptic potential, but also the precise timing of the release. That is the moment where the neuron transmitter has activated the postsynaptic side, and there is a response in potential. So using this multiple time scale approach, we have a way into calibrating well and capturing this delay quite precisely. We can also compare the model with data from excited free synapses. So this one was extracted with permission from a paper by Thomas Sudoff and collaborators. And here as well, there is a specific delay at each synaptic event, which we can capture using our approach. Okay, so that was it for my part. I'm still going to give you a little bit of summary and some perspectives. The main idea that we wanted to promote is this, with the example of the delayed transcritical bifurcation, is really the idea of bringing a sort of toolbox towards synaptic transmission models, and that's the toolbox of multiple time scale dynamics. So all these experimental work has shown that there are indeed many different scales, particular time scales, involved in these different modes of synaptic transmission. And so we felt that it was quite natural to invoke these multiple time scale dynamics in the modeling. And we think that, at least to the best of our knowledge, it had not been used in previous modeling approaches. So we came up with a simple model. So again, we try to keep it parsimonious to have a sort of proof of concept. It's not perfect at all. There's a lot of things missing, but at least it kind of unifies in a rather compact framework the different modes of exocytosis, of neurotransmitter release, and short-term synaptic plasticity. In particular, we can obtain asynchronous release related to the data I've shown you before, but also synchronous release, where the delay to release will be in a sort of more normal range, so to speak, by adjusting the configuration of the P1, P2 model and its parameters, and also spontaneous mini release. So we have different ways to do that, but one particular way is to shift these two states away from this region, make sure that they are quite close from each other in such a way that by adding some noise in the P1 equation, we would spontaneously enter into this loop, which we could do also with some periodic deterministic dynamics. We have tried to motivate and justify, at least partially, the forms of the equation using this cell cycle model consideration. And we see that as a sort of alternative to other approaches, for instance, hardwiring the delay as a time delay in the equations, which is possible, but might make it more difficult to obtain variability in this delay, and also ends up with a quite complex mathematical object. There are also other approaches with stochastic models, but that is, again, a slightly different approach that we wanted to take, where we would obtain the delay as the result of dynamic processes between slow and fast elements here, these different protein complexes. In terms of perspectives, we have some predictions made by the model, and they are related to some postulates that exist in the literature about what subgroup of protein could be a good candidate to organize the transition between different modes of release, in particular complexin. And we have ways to justify this with our model, but it needs to be further tested with experiments. We're also trying to extend the model currently by capturing further multiple timescale dynamics in the other part of the vesicle cycle, the so-called endocytosis, where the vesicle is reawakened by the cell Henri field. But there are known differential processes, some faster than others, to where this release can occur. So that calls for an extension, and still this slow-fast framework could be pertinent. In fact, we have a new postdoc in the group here in Sophia-Antipolis, Mathieu, that is working on this aspect. And of course, we would very much like, and that's what we're also starting to do, to connect with the recent results by Afia and include this multi-timescale synaptic transmission model into microcircuits involving both excitatory and inhibitory neurons, and study key phenomena, in particular this hyperexcitability that she has mentioned in link with Alzheimer's disease. Okay, so if you're interested in knowing more about this work, this is the paper, and I would like to mention and thank my collaborator, Serafim Rodriguez, Martin Krupa, Jesus Cortez, Terry Senowski, and Afia, of course. And a special mention for Serafim, who is really the one who had to dive into the rather complex literature on RSM interaction and the work by Sudov. So hats off to him, and thank you.
Thank you, Mathieu and Afia. Questions or comments?
I see the yellow hand of Bruno.
Hello, thank you, Mathieu and Afia, for this nice presentation. I have a question for Afia. Okay, sorry. Afia, I've heard that in Alzheimer's disease very early, there's a microphone, I guess.
Right here? I think it comes from there, but I'm not
Maybe
sure.
it's, I don't know, my foot. Hello again. So I would like to know, in the early phase of Alzheimer's, sometimes you have these monomers of tau oligomers that go in the presynaptic button, and they stick to the vesicles. Okay, and when they stick to the vesicles, they prevent the vesicles to be released properly in high frequencies. And this, I believe, that will cause a decrease in the release probability. And I would like to know how this can impact in the hyperexcitability that you say that can lead to synaptic loss.
It's a really interesting question. Thank you for this one. So the blockage, is it? Do you know which type of synapse it's at? Is it a glutamatergic synapse, or is it the identification? I mean, we have started to look at tau and the interaction of tau and amyloid beta. But one thing for sure that's been published is that tau has little effect on hyperexcitability. It's really amyloid precursor protein, or APP, that is required for the hyperexcitability. So that's the only, sorry if I'm not being so helpful, but tau is not so commonly known to be associated with hyperexcitability. But in the new model that we haven't developed it, that we crossbred and taken Seoto's work. So now we have a really nice model of A beta and tau. So we will use this to actually investigate. So watch this space, that's all I can say. But I know for sure tau has little effect on hyperexcitability. That's a presynaptic terminal.
You want to speak louder?
Okay. What will be the effect of tau in the dynamics?
In the dynamics of synaptic behavior? Something we haven't looked at, because the model that we've only really studied to in detail, and again, this is over the last 5 years, is the APP model. And there haven't been that many people in the field to really investigate the network circuitry. You know, probably from the literature, that people who work on tau or amyloid beta are really looking at the molecular side. Only the cellular circuitry aspect it's come fashionable at the moment. We move to the field to answer some of these unexplained problems. But to answer your question in short, I don't know is the answer. So something it's to definitely look at. But definitely it could be that it degenerates, could be to do with targeting specific subsets of inhibitory cells and allowing maybe the upregulation of other types that causes a shift. So one study that we've done is that some inhibitory circuitries actually become stronger in Alzheimer's disease. So instead of, well, why isn't this helping hyperexcitability? No, because this circuitry is only designed to control inhibition. So if inhibition or disinhibition is enhanced, it's going to ultimately affect your excitation level. So there's a lot of evidence suggesting that excitability is being targeted by different angles in order to result in the hyperexcitability. And this can start really early onwards, even before the pathology develops. So even before the amyloid beta plaques form or even tau is seen in some models. So, and this could be more linked with presynaptic proteins such as the Wnt signaling I mentioned. So I hope that helps. Sorry, I can't reveal more because it's just unknown at the moment.
Okay, thank you, Afia. There is a question by Bruno online, I think. Could you?
Hello, can you hear me?
Yeah.
Yes. Hello. Thank you for this very nice talk. I have a question to Mathieu. When you have shown pseudo data in the comparison with your model, we have seen that in pseudo data there are several rebounds, some kind of rebounds in the response that you don't observe in the model. I wanted to know if these rebounds are important and if it is possible to obtain them in the model by maybe tuning parameters. So, do you see what I'm speaking about?
Yeah, now I see what you're talking about. It's well observed. You have an eagle eye. So no, we have not captured that with the model. You could indeed consider that these are fluctuations that could be captured with some added noise, or that these are oscillations that may be meaningful, in which case maybe some additional timescale could capture them. But as such, the model doesn't capture that.
Is it important? On the biological side, you don't know? I
don't know. Okay. Maybe Afia as a clue, but I don't.
Okay. Thank you. Anyway, it was a very nice talk. Thank you.
Thanks.
Thank you. Other questions or comments in the room? Let me just check on internet. No. So let's thank our speakers again for this great talk.